Good morning, everyone. My name is Dunn. I'm an environmental scientist with the State Water Board, as Eric already mentioned, and co-lead of the Freshwater Harmful Algal Bloom Program. Marisa Van Dyke is the other co-lead of the program, and we have a lot of overlapping responsibilities as co-leads, but generally, Marisa's with the Swamp IQ unit, which I'll talk a little bit more about in a second, which is a lot of the lab analysis and data management, and then I'm more of the programmatic lead of the program. We both work within the Office of Information Management and Analysis in the Surface Water Ambient Monitoring Program. All right, let's see if I can get through these slides. Okay, so before we go into the overview of what we'll be talking about today, I wanted to briefly introduce who or what SWAMP is, what we do, and why we're involved with HABs. So SWAMP works to provide water quality resources and information to both decision makers and the public about the condition of California's water bodies. The SHAB program is part of a somewhat recent, within the last 2 years, initiative to address this growing issue of HABs and to support the protection of animal, human, and ecosystem health. The program also provides outreach and technical support services to a broad audience, so both decision makers and the public. All right, so today we'll kick things off with the general overview of cyanobacteria and HABs, which Eric briefly touched on. We'll go into a little bit more detail. And then I'll talk a bit about the SHAB program's guiding framework, the HAB assessment and support strategy, and then I'll hand things off to Marisa, and she'll go into detail about the key components of the strategy and some of the resources that we have available through the California HABs portal. All right, so what are harmful algal blooms? Algae and cyanobacteria have been around for billions of years, as Eric mentioned, and they're natural components of both marine and freshwater ecosystems. They perform many roles that are vital to our aquatic communities, including being a food source and producing oxygen. Cyanobacteria are often referred to as the pioneers of life on Earth. However, when conditions are favorable, algae and cyanobacteria can rapidly multiply, causing these blooms. And it's important to note that not all blooms are harmful, since only a small number of algae and cyanobacteria species are capable of producing toxins. And I'll talk a bit about the toxins in the next slide. HABs occur when colonies of algae or cyanobacteria that are capable of producing toxins rapidly grow and accumulate in the water column. HABs can impact human, pet, livestock, and wildlife health. They can occur both in freshwater lakes and rivers, such as those shown here on this slide. So to the left photo there, it's a bloom in Iron Gate Reservoir on the Klamath River. The center photo shows a bloom in the South Fork Eel River. And as you can see in this picture, this bloom originated in the substrate, or benthos, which is referred to as a benthic bloom. And then the photo on the right is a bloom in Lake Oroville. So as apparent by these photos, blooms come in all different shapes, sizes, and forms, and are very diverse throughout the state of California. All right, the animations are a little weird, so I'll just click through them. So a bit about the toxins. We refer to cyano-HAB toxins as CyanoToxins, and each of the cyanotoxins produced have different health effects for people and animals. The four main categories of toxins are dermatoxins, which affect the skin. These symptoms usually show up as skin rashes or allergic reactions. Hepatotoxins, which can affect your liver, and cytotoxins, which affect the kidney. And then the last main category are neurotoxins, which impact the nervous system. Neurotoxins are extremely fast-acting and can lead to paralysis and seizures. Cyanotoxin effects on domestic animals include vomiting, convulsions, and sometimes death. What we often see in California are effects of neurotoxins on dogs. They're especially susceptible because they will preferentially go for cyanobacteria. They're attracted to the stinky smell, and then when they get out of the water after playing, they'll lick their paws and fur, which results in ingestion of cells, and toxins are released as those cells lyse and break open. So essentially, they have a higher risk of exposure to the toxins. Human health effects include rashes or flu-like symptoms, vomiting, and sometimes seizures. It's important to note that boiling the water does not remove toxins. It actually breaks open cells and will release toxins present in the cell. Also, many conventional water treatment systems don't remove toxins. And as part of our program, we've been working with the State Water Board's Division of Drinking Water. So when we become aware of a cyanotoxin or HAB issue, we make sure that DDW is alerted so that they can respond as appropriate. And finally, wildlife effects. Cyanotoxins have been found in fish and shellfish organs and tissue where they bioaccumulate. Research has shown that the deaths of at least 21 sea otters back in 2000, I want to say 7 or so, were linked to the cyanotoxin microcystin. And those reported deaths were in Monterey Bay. And what the research showed was that they consumed shellfish that had accumulated cyanotoxins, and the cyanotoxins had originated in a freshwater lake, and the toxins were transported down into the estuary where the otters then ate the shellfish. All right, so a bit about the different cyanobacteria, some common HAB-forming cyanobacteria here in California. The different genera produce different toxins. So this slide is just showing some of the common benthic cyanobacteria on the left of the slide found in rivers. And as I mentioned earlier, these genera are found growing on the substrate or benthos of rivers and sometimes reservoirs as well. So Formidium can produce 3 toxins, one of which is a hepatotoxin, microcystin, and then both anatoxin A and saxitoxin, which are neurotoxins. Anabena is also a benthic bloomer, and it produces the same toxins as Formidium with the addition of cylindrospermopsin, which affects the kidney and the liver. And then to the right, a couple of the common surface bloomers in lakes, Aphanizomenon and Microcystis. I won't read through all the toxins here, but you can read for yourself. The message here really is that it's important to know what species of cyanobacteria are present within a particular system so that you can then determine what toxins may be present and what the potential health risks are, and then to be able to communicate that risk with the public. Okay, so this slide is getting across the point that it's important to understand the risk factors for water bodies that contribute to the likelihood of HABs. So those risk factors, this was taken from a paper by Hans Perl and Tim Otten. The risk factors are listed on the left-hand side of this table. And HABs are more likely to occur in warmer months and in warmer or slow-moving water. It's thought that climate change is acting as a catalyst for producing blooms. Nutrient over-enrichment is also considered a factor in bloom formation. And these risk factors are manifested in part by these cofactors that you see on the right-hand side of the table, which can then lead to biostimulatory conditions. And many of you may have heard this term. If you're a policy person, we have a biostimulatory substances policy that's coming out, and it's getting at just this thing. Or these conditions are basically conditions that are optimizing aquatic growth, including the growth of cyanobacteria. The take-home from the slide is that the alteration of watershed conditions in a way that would cause warming or slowing of the water, plus the addition of nutrients and other pollutants, are contributing to conditions that promote HAB growth or aquatic growth. So this slide is a summary of data on reports that we received last year the year before last, so 2016 is compared to 2017. And the take-home is, we had 91 reports, and Marisa will talk more about, if you guys are wondering what are reports and what are advisory signs, Marisa will talk about that in a second, but the number increased twofold from 2016 to 2017. So this is a growing issue. We're still collecting data on, what's causing this, and if this is in fact an increase in blooms or if it's an increase in awareness. I don't know if we can draw any sound conclusions at this point, but the fact is they are increasing, and the resources are increasing. Okay. So this slide is just a closer look at 2017. I'll just briefly touch on the advisory signs. So these signs are posted when a bloom is reported and we have some information about the toxin levels that may be present, or based on visual indicators that show that there may be a bloom. So the caution level, we had the majority of the advisory postings were in that caution level, nearly 100. And this is either low toxin levels, or caution signs sometimes go up as we're investigating a bloom. Dogs are not allowed in the water at caution, and the sign also cautions folks to keep their kids out of the water and livestock out of the water. At that warning and danger level, we had 42 advisory postings. The warning level is the water body has been closed for body contact, and the danger level is it's closed for all recreational uses. And then this kind of boring slide, no pictures, but this is just summarizing human and animal illnesses that reported in 2017. Clear Lake had one of their largest fish kills, which severely impacted sport fishing. So what fishers are reporting is that there's been more smaller fish and less of those big prize bass fish, and they have these world-renowned bass tournaments. And then also down in a few lakes in Long Beach, there were reoccurring fish kills. Okay, so with all of that, and this growing issue of HABs, and now that everybody's familiar with the risks that they pose and the data showing that they are increasing statewide, I'll talk a bit about what the state is doing to address this growing issue. So this strategy was written, I think it was put out in 2016, and it was in response to this growing occurrence of HABs in California and worldwide, really. You know, we've been seeing more severe and prolonged blooms in many water bodies across the state. Lake Elsinore, for example, is in bloom year-round, and there's multiple toxins present. I think there's 4 different toxins in that water body. As the previous slide showed, we tracked nearly 200 reports of blooms last year, and HABs are a significant concern for water quality and can impact a number of beneficial uses, including contact rec, drinking water, aquatic life, wildlife, so on. And they also pose risk to public health and wildlife. So together, these concerns drove the formation of the statewide strategy. And because of the diverse impacts to ecosystems, recreation, drinking water, fish and shellfish consumption, our strategy involves a multi-agency and stakeholder implementation approach. So SWAMP works collaboratively with CCHAB, who I'll talk about in the next slide, to implement the overall strategy to address HABs in California. For those that aren't familiar with who CCHAB is, it's the California Cyanobacteria and Harmful Algal Bloom Network, CCHAB, and it's utilized to build statewide partnerships and implement components of the strategy. The network consists of diverse participants, and their knowledge is a key resource for this effort. CCHAB is a platform to identify data gaps and monitoring of HABs beyond microcystins, which is a common cyanotoxin throughout much of the U.S., but here in California, we have multiple genera producing multiple toxins. So the take-home here is that the CCHAB network is the key to strategy implementation and in addressing the causes and impacts of cyano blooms in California, and it's also a plug to join the CCHAB network if you're not already a member. You can contact Marisa or myself if you're interested in learning more. Okay, so a couple more slides and I'm going to hand it off to Marisa. This is the main pieces of the strategy framework. Like I said, about a year and a half, 2 years ago, this assessment and support strategy came out, and it outlines actions and tools that will help address the increasing incidence of blooms throughout the state and help us to learn more about the conditions and triggers leading to blooms. The diagram here outlines the framework described in the report. And the strategy has 3 main components, which are response to HABs, ambient monitoring, and risk assessment. And then those are divided into water body scale and regional or statewide scale. At the water body scale, this is immediate event response, This is where the majority of our swamp resources are tied up during the busy bloom season in the summer months and beyond that, we're finding. But, we're coordinating bloom response, including identification, working with the lab for sampling and analysis, providing assistance with posting of advisory signs. Marisa will talk to those shortly. Long-term event response, this includes the development of local action plans, TMDLs in some areas, and implementation of management and remediation strategies. And the strategy also calls for regular monitoring of water bodies with recurrent blooms for those at high risk for blooms. At the state or regional scale, the strategy calls for development of infrastructure to support response to bloom events. And again, this is where we'll be talking in detail in the next few slides, and again, where we put a lot of our resources. And then monitoring on a larger spatial and temporal scale, which consists of adding cyanobacteria and toxins to ongoing monitoring programs. And lastly, to assess which water bodies may be at risk for blooms that would lead to regular monitoring. Okay, so as I mentioned previously, our primary focus has been building the program infrastructure for the parts needed to support the strategy. There are 7 main components that we're putting the bulk of our resources towards. I won't read all of those. You can see them for yourself here, and Marisa will go into those details now.
All right. Thank you, Ellie. This is Marisa Van Dyke, and I'll continue the content here today. First off, one of the primary components of the infrastructure is a centralized website. So under the California Water Quality Monitoring Council portals, you'll find the California HABs portal. It serves as a centralized website and is essentially a one-stop shop for all things HABs statewide. It's used to disseminate the resources that are created, such as trainings, standard operating procedures, event response guidance, and many other resources. It's also a key tool in outreach to stakeholders and used for HAB incident tracking statewide. On the landing page here that's depicted on the slide, left-hand side shows the interactive map that tracks voluntarily reported blooms. And then by clicking on it, it also has downloadable content. The right-hand side of the web page provides quick links to resources. The top toolbox has our most visited resources, including Report a Bloom in red. And then the lower section has many different resources and answers to common questions. And if you just want to click on the animation there, we just want to highlight that we have a component highlighted that goes directly to the drinking water web page created by the Division of Drinking Water, and it provides local contacts at each of the drinking water's offices, as well as information on existing state and federal guidelines, tap water testing, cyanoHAB treatment, and health impacts. So please check that out if you're interested. And before we move on from the landing page of the website, I want to highlight the reporting of a bloom and what that entails. So in the top of the toolbox, we highlight that kind of that key tool. So the public and resource managers are encouraged to report potential HABs and any related illnesses to the state's reporting hub by using any one of the following kind of tools, either filling out the online reporting form, calling a toll-free number, or emailing a central inbox. Others, these reports triggers a multi-agency coordination effort to respond to current HABs and to track the frequency, distribution, and impacts of HABs in California. All right, and going back to the infrastructure, one of the second primary components of it is the need to create HAB incident response guidance. These products have been developed by participating agencies and partners in the CCHAB Network. Ellie just clicked on all the animations for you. It's all right. So the top table shows one of the key components of the guidance that includes thresholds or triggers for water monitoring and posting health advisories. These thresholds are based on toxin concentration and biomass of the cyanobacteria. Again, that's the top table. The California Office of Environmental Health, Hazard, and Assessment, called OEHA for short, evaluated toxicological data and literature on animal and human health effects of 3 common cyanotoxins to develop these numeric triggers and created this tiered system for response. The thresholds are for microcystin, anatoxin A, and cylindrospermopsin. The image on the bottom of the slide shows the advisory signs. So when the thresholds are met, they result in the posting of signs in either caution, warning, or danger levels. The content of the signs were field tested by the California Department of Health and developed in 2 languages. On the right-hand side, we're showing just a snapshot of the decision tree. This is a key component of the guidance that steps the user through all the steps needed to respond to the bloom. This is written for the resource manager and local responders, including environmental health and public health departments. And this tells you a little bit more about how to take samples for testing, how to interpret the results, and when certain advisory signs are appropriate, and also when it's appropriate to depost signs at certain water bodies. On the central website in the toolbox, you'll also find a link to the field guide that includes standard operating procedures and pre-made forms for HAB incident monitoring. So the toolbox will take you to this, connecting web page. This field guide was developed by SWAMP, and it's maintained by SWAMP. It's formatted as an electronic document, so each of the sections and SOPs can be downloaded individually and just taken out in the field. They're kept pretty short so that you're not necessarily downloading a 100-page document, that kind of thing. It's made for use in the field. And the content is developed for a broad audience and different expertise levels, and also includes SWAMP's data quality objectives to ensure that we're collecting consistent data statewide. Please take a look at that and look a little bit more into standard operating procedures. State Water Board has also provided field sampling kits to all the regional offices around the state to assist with sampling at the local level. This support is triggered by voluntarily reporting a bloom, as we had previously described. These kits include a cyanoscope kit with field microscope and plankton nets, also tools for collecting water samples. It has fluorometers to measure different pigments created by cyanobacteria, and field test strips for testing one of the more common cyanotoxins called microcystin. We also have laboratory resources. The State Water Board has limited funding at a laboratory to help support cyanotoxin testing and taxonomy. And this information can then be used within the decision tree to decide on appropriate advisories at water bodies. And also under the field guide, you'll find a California laboratory list that compiles all known nationwide laboratories, not just statewide, and all the analyses that they can do. Just kind of helps everybody get started. In addition, on the webpage, you'll find several resources for education and outreach. This provides a snapshot of the available trainings and other educational resources that we have. It shows that we have a recording of the one-day workshop on identifying and responding to cyanobacteria. This was taught by Wayne Carmichael in 2016. And we also have recordings of the 3-day workshop that was hosted by US EPA and the SWAMP in spring of 2017. All right, on to another one of the components of the infrastructure is improving research and monitoring tool development. First off, under satellite imagery, California is a pilot state under the CYAN project, which is managed by NOAA and NASA to produce satellite imagery for tracking HABs, particularly cyanobacteria blooms. SWAMP is developing a web-based tool for viewing the satellite imagery. This will show about 250 of the largest lakes and estuaries in California, and the web page is currently under construction. They'll also have historic data back to 2002. so you can see a little bit more status and trends. And we're also working with researchers at UC Santa Cruz to develop satellite monitoring tool for smaller and narrow water bodies so that we can cover more of California with satellite imagery. In addition, SWAMP has funded several other research projects that will be launching this spring. This includes development of passive sampler devices, potentially SPAT or other kind of passive sampling devices for bloom surveillance, improving benthic bloom monitoring and laboratory analysis, and also to validate quantitative PCR methods to detect the toxin-producing genes in the water samples. All right, and this slide has some animations just kind of showing you on the portal that there's a lot more resources that we couldn't cover today. It has information about a CDC database that is being used to track HABs illnesses nationwide. It has many other trainings and collaborative kind of tools there, and the latest information on webinars, workshops, and other collaboration opportunities, such as CCHAB Network. All right, so I'm just going to step away a little bit from the HAB portal. We just want to highlight an assessment that we had done last bloom season, and we'll be repeating it again this summer and looking for partners to collaborate with. So prior to the Labor Day weekend last year, the water boards coordinated an assessment, and this was triggered by numerous requests from the public. They were asking if a local lake in their area was safe for their kids and pets prior to the upcoming holiday. So we reached out to the CCHAB Network partners that we knew were already monitoring around the state. About 8 organizations participated. And then to fill in some extra data gaps at popular recreation lakes, we funded regional boards to go out in the field and conduct some additional monitoring, collect those samples, and get them analyzed. To communicate these results to everyone, we created an interactive map shown here on the right on the slide. In summary, 43 water bodies or sites were sampled, and each of the colored dots here represents about 100 sampling locations, and they're all color-coded depending on these advisory levels. So about half of the advisories were at no advisory, so nothing to report. 33% were at the caution level, and 17% of the sites had the recommendation of no water contact, either at warning or danger levels. Before concluding today with this presentation, we have a quick note just to highlight some of the necessary permitting prior to managing blooms. The common short-term algae bloom management strategies include the application of algicides and herbicides in waterways. Many of these chemicals and other control practices are covered by the existing permit framework that's under the NPDES permit, either the statewide general or the individual. So please take a look at the website link that we've had on the slide. This will provide you with more details on enrolling into this permit and the process for it. All right, thank you for participating in today's webinar. Please feel free to contact us with any questions or comments that you have after this. It looks like we have a little bit of time to answer some questions. Eric, we'd like to ask if there's any comments submitted in the comment box.
Okay, I have a question. How do you get set up to voluntarily report algal blooms and then get access to regional sampling?
All right, that's a great question. Ellie, can we go back to that one slide that we have information? Sorry, we have to go through all the animations to get to it. Just like to provide you with information straight here. So on the California HABs portal, up in the upper right is that toolbox, and you can just click there to report a bloom, and it'll give you several options just to make it a little easier. There's not much setup. There's no downloads necessary. First off, it just depends on what kind of information you have. So if you would just like to do a brief, quick report, you can just call the toll-free number, or you can send a brief email to that inbox. However, if you have a little bit more information about what you're seeing and you'd like to provide some more content, we encourage folks to fill out the online instant form. So when you go to that web page, you'll have a link to it, and it'll send you just to this form. There's only about 7 required questions before submitting it. So, there's not too much that you have to fill out to get that going, but we do encourage folks to do that, because then you can tell us a little bit more about where you're seeing this bloom, if you've experienced any illness, or if there's any wildlife impacts. And then it'll also give you a tracking number after you submit that form. So then you can use that number to then correspond with anybody in the kind of reporting hub that manages the inbox.
And I just would add, too, that the sampling we have limited funding for it. So it's just for that initial response. Sometimes we have enough to do follow-up monitoring, but just FYI, it's for that first report to go out and take a sample.
Would you like to touch on the Bloom Watch app?
Yeah, sure. So there's a Bloom Watch app that we also, I don't think we have the link here, but if you go to the Report a Bloom on the portal, there's an option to use Bloom Watch. And so it's basically just an app you download. It's available for Android or your iPhone, Apple. And you can snap a picture of it, and that picture is shared. It's a community-based online community that can help identify the bloom and help track that information. California's been using it. I think we're one of the more active users of the app, and it's still somewhat under development, so just use it with caution, but it is a great tool. Easy to use.
Hey, we have another question. What's the ETA on the satellite tool?
Great question. So the satellite tool, we're actually meeting with some folks from NOAA next week, and some UC Santa Cruz researchers to get trained on a radiometer tool. So this summer we're doing a field verification study of the tool. The data is provisional and it needs to be ground truthed. Once we do that, I think we will have the tool finalized. I'm saying this tentatively, but by this summer. It is available in a beta version now. You can take a look, but just under the disclaimer that the data is provisional, it's still being ground truthed. But yeah, it's there now. Contact Marisa or I if you'd like that beta version.
Another comment came in. So a procedure would seem to have a reporting tool locally, report to SWAB, obtain field kit for verification. How do we monitor on an ongoing basis and determine the risk is gone?
Okay, let me just clarify something about the reporting. So you just take a look at the web page and report the bloom, and then someone would follow up with you, because all the reports are routed to a central hub. This is located at the water boards, and then we'll coordinate with the local or regional water board and other agency partners to address that incident. And depending on what's going on, if it's considered a potential bloom to go investigate, we'll work with the regional board folks to do that initial monitoring with their field test kits. So it'll be worked with the regional board staff. They'd work with the local water manager or recreational land manager to do that investigation. And so you'd want to report the bloom and then wait for communication from the local office. And then to monitor an ongoing bloom and determine the risk levels, that's where we point folks to the decision tree that's in the statewide instant response guidance document, and that's available on the HAB portal. It steps resource managers through all the steps for when you got an initial bloom report down to steps for lab analysis, when to post certain advisory signs, when to take down advisory signs, kind of like that cycle and recommended frequency for monitoring. So please take a look at the decision tree. It's a short one-page document, and then it has a narrative that kind of walks you through each of the steps, especially for first-time users.